to the CIBM Center for Biomedical Imaging Breakfast in Science seminar series. I'm very pleased to introduce Giulia Preti, who is a research staff scientist at the CIBM Signal Processing EPFL-UNIGE section. She will be sharing with us how decoupling of brain function from structure reveals regional behavioral specialization in humans. Thank you, Maria Giulia. The floor is yours.
Thank you, Pina, for the introduction, and good morning, everyone. As Pina said, I'm Giulia. I'm a research staff scientist as the CIBM Signal Processing section in Geneva. And in particular, I'm part of the medical image processing laboratory located at Campus Biotech here in Geneva. And today I would like to talk about a new way to integrate brain function and structure assessed with magnetic resonance imaging. So as you probably all know, one of the greatest advantages of MRI is actually that by changing the sequence of acquisition, you can assess very different types of information. One of it is brain function with functional MRI. In particular, we can retrieve functional activations in the brain, and we can look at how brain regions that are even far located communicate to each other. And another piece of information that we can retrieve is the structural one in terms of structural connectivity assessed with diffusion MRI. And in this case, we can actually reconstruct the physical connection between different locations in the brain in terms of white matter fiber bundles. And so we can look at how brain regions are actually connected by fiber bundles. So as you can imagine, these two pieces of information are very linked one to the other, as functional activation happens on top of the structural architecture underneath of fiber bundles that actually transmits the signals. And so what we wish for is to have an optimal combined technique, combined analysis in order to integrate these two pieces of information, so functional and structural. So before starting, I would like to spend a few words on these two techniques and see what are the elements that they yield and that we are going to work with. So in terms of structural information, diffusion MRI allows us to obtain what we call a structural connectome. So let's see how. Usually what you need is a brain atlas, so a parcellation of the cortex in subdivision into different regions that are usually given by a reference atlas. And you can put that on top of the virtual reconstruction of fibers in the whole brain, what we call tractography. And that yields a brain graph in which each node is simply the region of the atlas, and the edges between nodes are represented by a certain property of the bundles that connect the two regions. For example, in the simplest case, it can be the number of fibers connecting two regions. So this brain graph is what we call structural connectome, that here is visualized also as a matrix, and that we can consider as a bit the wiring diagram, as it tells us information about how the different regions are connected structurally in the brain. In terms of function, instead, as we said, functional MRI provides information on brain activity. And essentially, there are two types of experiments that we can have. One is task-based, so the subject is performing a task inside the scanner. And the second one, instead, is resting state, where the subject is basically not doing any task, but just resting in the scanner. And it's in the past couple of decades that this technique, resting state, showed his importance, as it was actually demonstrated that while you're resting, there is still a lot of activity going on in your brain. And in fact, all these co-activation, co-deactivation that you see are do not happen randomly, but there are actually very specific networks, very consistent across subjects, that are recruited during resting state, called resting state networks. So from here, the importance also of using this technique, which is quite simple to acquire, and from usually we retrieve the information about functional connectivity. So similarly as for the structural connectome, we can obtain a functional connectome, always starting from a parcellation. For our purpose, let's say the same exact parcellation that we used for the structural connectome. And in this case, instead, we can compute the average functional signal of all voxels inside each region of the atlas, and then assess the correlation, the temporal correlation between each pairs of region. And this metric is the one that goes inside the functional connectome, which is again a matrix regions by regions that includes instead correlation. So basically the information about temporal synchronization of different regions in the brain. That's why we can refer to it as a bit of communication diagram, as it tells us information about how different regions in the brain communicate. So now, how do we put these these two pieces of information together? So this was explored already a lot in literature, and as you can imagine, the very simple idea that comes to mind is simply to correlate functional and structural matrices, so the functional and the structural connectome. And this was explored at the beginning, and as you see, there is indeed a trend of a positive correlation between structural and functional connection, as we can expect. But that you see, it's not even that strong. Of course, we can imagine that all the complex mechanisms that drive this integration between function and structure are hardly representable by a simple one-to-one correlation. In fact, also we have to keep in mind that the functional connectivity was shown to be dynamics and to change at a much faster scale than the structural connectome. So during the time of the acquisition, which can be around 15 minutes, we have to imagine to have a dynamic functional connectome that is changing on top of a static structural one. So this also needs to be accounted for and cannot be simply described by one average measure such the correlation. And so efforts in order to try to explain how the dynamical function evolves on top of a static structural architecture have been performed with a much more complex technique, which is whole brain computational models. These models try to simulate functional time courses based on assumptions on the structural connectome underneath and models of neural activations. And so in the recent years, these models showed to be useful in order to replicate the behavior of the brain under certain conditions and being able to modulate it. But at the same time, the downside of this method is that there are usually many parameters to tune in order to optimize the model that remain a bit of a black box to the user and that are not so easily interpretable, let's say, in order to meaningfully optimize them. So what we want to do today, what I want to propose you is a novel framework in order to integrate the functional and structural information, which is based on graph signal processing. Graph signal processing is an emerging framework that actually applies and converts all transformations and operations of conventional signal processing, but to signals that are defined on graphs. And this was only recently, in the past few years, experimented on the brain. So let's see a bit more in details. as the name say, we need a graph and we need the signal defined on top of it. And that's exactly what we have, because we do have a graph that in our case is the structural connectivity. And we have a signal which is the functional pattern of functional activations at each time point. And as you see, this signal is defined on top of the graph. And our question is therefore, how is the function using the underlying structure of the brain? A bit more into details, we have a graph that we can simplify like that as a bunch of nodes connected, which simplifies our structural connectome. And we have functional time courses that we can measure at the very same nodes. As I said, we consider the same parcellation so that we have these two pieces of information for each node. So our graph signal, the fMRI time courses at each time point, is defined on top of the graph in this sense, because it's a vector associating a value for each node. So we have to consider one specific time point, in this case, not the full time course. And you can see that by considering one time point, you associate one value, which is the bold activation level at each node of the structural graph. And then you know from the structural graph how these nodes are connected to each other. In our case, the graph evolves in time because we have functional time courses, but it's important to understand that the analysis that we do is not in a way in time, is not a temporal analysis, the graph signal processing one, I mean, but it's a spatial analysis in the space of the graph, because we repeat then the analysis for each time point, but we act at the level of the functional pattern at one time point. So to start, I would like just to here briefly introduce all the steps of the pipeline with a bit more technical and mathematical details, and then I will explain it more conceptually into details. So what we do is first computing a graph Laplacian from the structural connectome, and then eigendecompose it in order to find the eigenvectors U of the Laplacian of the structural connectomes. We can refer to these as structural components, which form a bit the backbone of our structural connectome. And what we do next is taking our signal, so the functional pattern at one time point, and try to explain it in terms of this structural component. So see how much each of these structural component is included in the functional signal at one time point. So this is the equivalent of performing a Fourier transform in conventional signal processing, but it's what is called graph Fourier transform, because it transforms our fMRI signal from the domain, from the spatial domain, the nodal domain of the graph, to the spectral domain of the graph, which is spanned indeed by the Laplacian eigenvectors. So let's see a bit more into details what it means and what we can do with this. So as we said, we start from the structural connectome and we obtain structural components. So in literature, these are referred to also as structural harmonics, and we also call them that way. They look like this, and we call them structural harmonics because each of these patterns, so the eigenvectors, are associated to a particular spatial frequency. And these spatial frequencies are represented simply by the eigenvalues of the eigendecomposition. So as you know, each eigenvector is associated to an eigenvalue. And we will see now a bit more into detail why they have a meaning of spatial frequency. So if you order them in increasing order, so starting from 0 and increasing, the first one will have 0 spatial frequency. It's a bit the DC of our decomposition. And as you see, it's a global pattern. So 0 spatial frequency means that you see there's no spatial variation. So this pattern is almost constant, let's say. But then there is a bunch of structural components, the first ones that that are characterized by low eigenvalues, so like low frequencies, that are characterized by having slow spatial variation. So these ones let's say here are some few examples, look like this. So they are global components, global patterns, in which the spatial variations are very slow. You see there's no variation from red to blue in nearby nodes, but they capture what are the main directions of the structural graph. For example, here you see the difference between left and right hemisphere, or posterior anterior, and so on. And so by construction, these eigenvectors are obtained by optimizing smoothness on the graph. It means that for these ones, the very first ones that optimize this criterion, they are very smooth, very attached to the graph. What it means is that two nodes that are very connected in the structure will have a very similar value in these patterns. And so therefore, what we can say about these patterns is that they are very coupled to the structural connectome. So there is a lot of information about the structure in these patterns. On the opposite, if we look at the other side of the spectrum, so the high spatial frequencies, we see that we have patterns that are very different. So first of all, they are scattered usually. And variations are very fast, spatial variations. So usually you have nodes that can be also nearby that go from blue to red, so they change sign. These spatial variations do not reflect much of the structural architecture. So these subdivisions into patterns, structural patterns that are more attached to the connectome and patterns that are less is key then to what we're going to do next. So trying to yield a metric of coupling with the structure, function structure. But let's see. So next, we consider a functional MRI frame at one time point, so our graph signal. And what we do is trying to explain it as a linear combination of the structural harmonics, of the structural components. In other terms, we want to assess the contribution of each of the different structural components to the functional signal in these coefficients as hat1, as hat2, and so on. And so by doing that at every time point, we can actually, so the set of structural harmonics doesn't change, but what it changes is the estimation of the coefficients that put together these structural components to form the functional signal in time. So we can see at different time points how differently the structural components combine in order to give origin to the functional signal, basically. And then, if you remember the subdivisions that we have in low frequency and high frequency components which are attached or detached to the structural connectome, what we wanted to do here is to filter the functional signal, so to decompose our functional patterns into two components. One that only includes, so it's just the linear combination of low frequency patterns, and one that only includes high frequency patterns. So the first one we know to be coupled to the structural connectome. The second one we assume it to be decoupled from that. To give you a visual example, this is a real example of one frame. It's one subject at one time point, fMRI signal, and the fMRI signal will be split into two portions, and then if you sum them up, you get again your original signal. A coupled portion that only includes low frequency structural harmonics, and a decoupled portion. And you can see very clearly here that in the coupled portion, you get patterns that are much more similar to the structural architecture, and in the decoupled portions you have somehow the activity that rides on top of it. So for example, you will have, if you have asymmetric activations in the fMRI frame, they will be much more probable to end up in the decoupled portion, as they are probably less explainable by the structure. So we do that for each time point. So for each time point, we split the fMRI signal into two portions, a coupled one and a decoupled one. And then what we asked ourselves is, are there regions in the brain that actually most of the time, so in general across time, are more coupled or more decoupled to this underlying structure. And so we computed the norm across time of these two portions of signal, the coupled and the decoupled one. And this gives us a global measure of coupling and decoupling per node, so per region across time. And then we compute the ratio of this decoupling over coupling. And this is what we call structural decoupling index that we obtain per node. So it's a regional measure of coupling of function to structure. And we did that for applying it to 56 subjects, healthy subjects of the Human Connectome project during resting states. And this is the results we get. So what we can notice is that actually the structural decoupling index, so the coupling structure function, has a very specific distribution in the brain, very specific pattern. And regions that appear to be more coupled to the structure, the ones that you here see in blue, belong to mainly primary sensory networks like visual, somatomotor, auditory, while regions that are more decoupled, so you can see here in red, belong to higher level cognition in terms of function, so memory, decision making, and so on. And actually, this subdivision, so this gradient present in the brain, was found to be consistent with many other techniques that have nothing, that didn't explore structure-function coupling. And one interesting one is the strong congruence with genetic studies that show a very similar subdivision, but based on the proteins that are used for neural transmission and basically in genetic expression. So what they found is that the network that for us appears as coupled, so the one in blue, was found to favor the use of genes for faster response, so do have a faster response. While the network that is decoupled for us was found to use gene that favor a slower, more sustained activity. And this fits very well with a possible interpretation because we can, in more easy terms, we can think, for example, if we have a stimulus of the primary sensory cortex, for example, we see a danger, we need to act fast. So what we know from the genetic studies is that the response will be faster. And what we are adding in terms of structure function coupling is that the functional signals then is more probable to use directed structural paths. So it uses actually the structural connectome underneath. It's like taking the highway, and it's probably faster. While if you are doing some higher level cognition tasks, like thinking about the meaning of life, thinking about your past, probably this doesn't require the same temporal limits, and so you can afford a slower response. And in terms of coupling, basically you have your functional signal can afford using, also being more liberal from the structural connectome, which might mean using undirected paths, basically. And then what we did was showing that the structural decoupling index has a behavioral relevance. So it's a bit justification of what we saw before visually, the subdivision into low and high, higher level cognition. And in fact, we just split the cortex into regions that have lower to higher structural decoupling index by subdividing into 20 percentile of SDI. And then each of these masks entered a meta-analysis in order to assess with neurosynth, in order to assess to which topic terms this mask would correspond more. And we actually found out that there is actually a gradient going from more coupled to more decoupled regions that spans behavioral domains from more lower level like motor, eye movements to higher level ones like memory, emotion, and so on. And this result was very consistent with the ones obtained by Margulies in 2016, which we wanted to replicate with the same technique, the same neurosynth meta-analysis, but which is based only on functional connectivity. So what they found was this same gradient spanning between primary sensory regions and higher level ones, but only in terms of functional connectivity. And what we are adding is that the same gradient is present also in terms of function structure coupling, basically. So to conclude, we showed here that graph signal processing can be very useful, and it's a promising tool to integrate brain structure and function, and that this structure function coupling depends actually on the specific brain location, changes with the network that we are investigating, and that it changes along a gradient with a specific behavioral relevance. So then as in the future, so what I didn't mention here is that there are different ways to filter the functional activity, so to define the filters. And of course, this can be optimized also in the future by introducing also different alternatives. And plus as future development, another thing that is for sure worth exploring is the temporal dynamics of this structure function coupling, which is explored by this method. I mean, we can retrieve this information, and the changes of this structure function coupling across these different conditions, so not only in rest, but also in task. So with this, I thank you, of course, all the members of MIPLAB, and you for your attention.
Thank you, Maria and Giulia. So I'd like to open the floor to the audience for any questions.
Hello.
Hello, Francois.
Yes. Thank you very much, Giulia. It's very interesting. I have just a simple question, just to start. How do you explain this mismatch between, I mean, this lack of correlation between structural and function? Is it due to the fact that some of the high cognitive function cannot be really tracked by structural connectivity because it's involved, for instance, deep nuclei, like you have listed the cognitive control, and cognitive control is a very complex structural, I mean, network involving caudate, perhaps insula, other regions. So my question is, do you think this decoupling between both is due to a lack of sensitivity, let's say, of the structural connectivity?
I think, I mean, I think that actually the structural connectome that was used here is quite advanced. So of course you have more difficulties in retrieving certain tracts, but it's the one obtained from the sequences of the HCP, and it was not included in this paper, but more recently we also use one that includes subcortical regions, so more deep structures, and the results basically do not change. So I think more that it's due to the fact that indeed these higher cognitive level functions are not so easily explainable by direct connections, and therefore they will kind of go into a more decoupled signal which has structural information, because of course the structural harmonics always come from the structural connectome, but less easily explainable by these global structural patterns. So yeah, more decoupled, more undirected paths, for example.
Just a little question, I missed the point. When you have this hub, I mean this structural image, and you have color, maps, and size of the hubs. Can you just repeat to me what represents the color and the size?
You mean these ones?
Yes.
Yeah, so these are, so it's just the structural harmonics, so the Laplacian eigenvectors. So the color is just positive, negative, blue, red, and the size is, I mean, here it's represented just the values of the eigenvectors. So the size is proportional to the value that is represented. So for each, so the eigenvectors will be of the Laplacian of the structural connectome will be these vectors of n regions in this case, and here we just represent them on a brain graph on top of the brain.
Okay. Okay. Thank you. Okay.
Giulia, I have a question following Francoise a little bit because it was not clear to me. The thalamus is quite important in managing, is the hub of many of the functions. I didn't understood in your connectome, do you take care of subdividing the thalamus in the different nuclei that we know a priori that they are in charge of different functions? Or you have the thalamus as a whole?
I think you have it as a whole. I didn't actually specify it, so you're right. The parcellation that we used for this is the Glasser parcellation, based on a paper in Nature in 2016 of Glasser, that divides the cortex into 360 regions. So 180 left, 180 right. I don't recall exactly if because some regions are included even of the subcortical ones already, but then we extended it by adding 19 more subcortical region where the thalamus is included, but I think as a whole. Yeah.
Because it would be nice to have as well, if you, I don't know the limitations in terms of spatial resolution, or I mean, this, it's not clear to me at which level of detail, but it would be interesting, of course, to just translate your analysis to the different nuclei. I think it would be very meaningful with your method, that it's amazing. Another quick question, just did you have the opportunity already to apply this to pathological data to see if exactly this is a dysfunction in a group of pathology like, I don't know, schizophrenia or
Yes, so it's on the way. So we are trying to apply it to different types of pathology. And yeah, so the interesting thing is that you can, so for example, schizophrenia, right, or stroke, or agenesis of the corpus callosum, for example. The interesting thing is that you can do different things when you have a group of patients. So for example, you can keep the same template for the structural connectome, so with the same structural harmonics, and you can project the function of your patients into it in order to see how differently they would use a set of healthy structural components. Or you can use and decompose structural of the patients, so they would have their structure and their function. Or you can actually also do the opposite, so see if you want to use their structure and how if a healthy function would use differently these pathological structures. So for now we are exploring different ways, and you have these interesting opportunities. Yeah, you will have for sure what I expect. You would see, you know already that you have some deficits depending on the pathology in terms of function, in terms of structure, but here you would have the opportunity to see what changes in the pathology also in terms of coupling, basically, structure functions.
That's good. Thank you, Giulia.
Thank you, Matt.
Great, thank you. Any other questions from the audience?
Hi, if there's time, I would like to ask a quick question. Hi, Giulia. So I really enjoyed your talk. I was just wondering how sensitive this method is to the underlying assumed structural connectome, because I mean, we know it's not really a ground truth, or it's not really set in stone. There are issues with false positives and so on. So depending on how much you clean up this initial connectome, how do your results change?
Yeah, so what I experimented is this, it's quite stable, it's quite robust, even the results, to some variations on the structural connectome. For example, subject-wise variation. So here in the method, what we do is taking an average structural connectome of the whole population so that we have a decomposition in structural harmonics that is common to everyone, and then projecting the function of each individual. But I didn't have, let's say, so it was quite consistent. I didn't have much changes by changing the structural connectome like group, let's say. So trying to average across other groups, or even taking one individual, let's say. I mean, I didn't try optimizing too much the pipeline to obtain the structural connectome. So this is obtained with spherical deconvolution on the HCP data, and it's with probabilistic tractography and using SIFT in order to SIFT2 actually in order to clean the connectome for false positives. So it should be accounted for. But the only requirements is that, I mean, you can also clean it in a different way, or have a matrix more or less sparse if you want to exclude, for example, small connections. But the only thing is that the matrix should remain suitable for then the next decompositions. So the problem with a bit less sophisticated connectome that I found is that then the Laplacian decomposition was not retrieved in a correct way, let's say. But other than that, for the data that I tried, also obtaining some in different ways, for example, DSI, or it was quite stable, yeah, in terms of these patterns of the structural decomposition.
Okay, thank you.
You're welcome.
Okay, thank you very much. We'll have time later to ask more questions if you like. So thank you, Giulia, for this very interesting talk.